Okay, thanks. Yeah, so thanks, Ulrika, and thanks so much for asking me to come and talk to you. I sort of have two hats. I'm a statistician, but I'm also a health economist. So I'm very interested in how we reflect uncertainty and evidence into economic models in order to help us make decisions reflecting that uncertainty. And so Bayesian methods actually are really, really crucial to that in my view. So thinking about parameters and how certain we are about them, but also then saying, well, can we make a decision now? Or actually, do we need to go and update our evidence by collecting new evidence to be able to then make better decisions? So it's a very natural Bayesian process, actually, this idea of updating your beliefs. Okay, so I'm going to start off by just telling you a little bit about what evidence synthesis is and what network meta-analysis is. I can't give you a very detailed introduction to network meta-analysis in this time, but some of you came yesterday, so you probably already had it. Okay, so what is evidence synthesis? It's where we're trying to combine evidence from multiple sources to then be able to get some overall pooled estimate of a parameter. And that's important when you're making decisions, because you want your decision to reflect all the evidence that's out there. You don't want to pick and choose your evidence to be able to, you know, if you then make a decision based on just one study, well, perhaps if you'd looked at a different study, you would make weaker picking decisions. So the idea is you want to be able to reflect all the relevant evidence. So this is a simple example of evidence synthesis, a simple meta-analysis where we've got various studies that have looked at comparing this combination therapy, this LABA plus ICS, that's inhaled steroids, and a beta receptor agonist, or against just using the LABA on its own. So it's looking at combination versus monotherapy here. And if you look at these studies, you know, you've got variation between them, which I can't point at both screens at once. So, but you've got variation between studies, but not a lot actually. Most of these studies are telling us roughly the same thing. And if you pull them together in a weighted average, then you get the pooled estimate at the bottom there, which has combined all that evidence. And actually, we're quite certain here that there seems to be more effective to use the combined therapy compared to the monotherapy, although there was one trial which found no difference between them. The majority of them did. But we can see that the width of that diamond at the bottom reflects our uncertainty in the pooled estimate. So we're much more certain having put all of that evidence together, so we can make stronger conclusions. That's what evidence synthesis aims to do. So where does evidence synthesis get used in the drug development process? Well, actually, it does actually get used all the way through the drug development process. Right at the very beginning, when sort of preclinical work's being done, there are people putting together studies on various different animal models and so on, to try and understand how to design the trials and to produce, you know, what sort of things to. I haven't done a lot of work in this area, but they're sort of trying, focusing more on the experimental design rather than making conclusions about effectiveness. So they're trying to sort of work out how we can take this forwards and then try and get successful possibilities to take through to phase 1 testing. At the phase 2 and phase 3 development of drugs, meta-analysis is used quite a lot. In fact, this thing called model-based meta-analysis, where they try to estimate dose response or time course models to try and work out what sort of dosing should be used, what time points should be collected in the big trials at the phase 3 stage, and whether or not to continue forwards with the development. But the bit that I'm sort of involved in is at the far end. So after a drug's made it through this process and you've got to a phase 3 trial with a very big study that's been done, then it goes through to licensing. So it needs to get licensed. It needs to be considered to be safe and effective to be able to be used in practice. And that's the first step. So once the study's got licensing, then in the UK at least, and certainly in other countries, I don't know, not all countries, but quite a lot of countries, there's a consideration not just of is this new drug effective, but is it cost effective? So we're not just talking about whether or not it's beneficial. Usually they've got this far, they are beneficial, but they might be very expensive. So we want to try and weigh up the costs and the benefits to decide whether or not it's worth using public money to be able to pay for this particular treatment, or whether that public money could be better used on other things which might give more benefits. So that's the basic idea of sort of reimbursement steps. That's that blue box there. And when you get to that stage, the question you want to ask is, how cost effective is this new drug compared to all the other options that we could use instead? So you've often got multiple treatments that you want to consider at that stage. It's not necessarily the treatments or the control and the intervention that's been used in their phase 3 trial, but actually you want to compare with all potential comparators. You know, we want to make sure that this is the best thing, it's going to be the best use of money for these patients. And network meta-analysis is a method which pulls together all the evidence available that allows you to compare multiple treatment options. And it's been used a lot in reimbursement because it answers the question that they need to answer, which is, you know, how effective is this treatment compared to all the multiple other options. So I do a lot of work with NICE in the UK, so that's the National Institute of Health and Care Excellence. And they're a reimbursement agency that do exactly that. They're trying to work out whether or not new treatments are worth it, whether they're value for money. And to do that, they use an economic model usually to estimate the net benefits. So the net health benefits minus the costs. And you monetize the benefits with a willingness to pay per quality adjusted life year. So that's how it works. But a really key input to those models will be the relative effectiveness of the multiple treatments that are under consideration. And that's what network meta-analysis can give us. So I mean, ideally, we'd have one big, large phase 3 trial on lots of patients that compared all the treatments that were. But in practice, we don't have that. Usually, the trials are just on 2 treatments, maybe 3 treatments. Randomized trials usually of new drugs often compare with something which isn't perhaps the best thing. It's just something which they can show is better than some old standard or even placebo, just to show that it's safe and that it's better than something. And that's what's required to get through the licensing part. But that's not enough to get you through the regulatory part. So there's a kind of a mismatch there. And I know that lots of people are trying to talk about how that mismatch can be bridged, but they're 2 quite different cultures. But there might be multiple trials making different comparisons, and some of those might be relevant. So the idea here is to try and piece together the evidence that we do have on the various different comparisons we do have to be able to compare the treatments we need to make our decision. So here's an example. This was a nice technology appraisal. Do I hide that thing at the top?
You just
Let's go. Apologies. Oh no, then now I've lost it in the big screen.
No. And then take this.
You hide it. Thank you. Sorry. Okay. I'm sorry. Yeah. Okay. Okay, thank you. So yeah, this was a technology appraisal which was looking at a new drug, empagliflozin, for treating patients with chronic heart failure with a reduced ejection fraction. So for this particular patient group, there was a trial that had been done and it compared empagliflozin against placebo. But what was used in practice was another drug, dapagliflozin. And so the decision question was empagliflozin versus dapagliflozin. It wasn't placebo because you wouldn't give patients nothing, you wouldn't give them placebo. So we needed to make that comparison. But there was another trial, the DAPA-HF trial, which had compared dapagliflozin against placebo as well. And so we can try and pull together that evidence to be able to make the comparison that we want to. And it's known as an indirect comparison. So dapagliflozin can be empagliflozin can be compared with dapagliflozin using this relationship here, that if you've want to make a comparison between treatment B and treatment A, well, we can take the C against A effect and the B against C effect and minus 1 from the other. Okay, so it's basically if you had one was 5 units better and the other one was 3 units better than placebo, then, well, the difference between them is just 2. So you'd expect that to be the difference between dapagliflozin and empagliflozin. And that's effectively what this method does. There's nothing very exciting, nothing very clever about it. It's just a simple sum. The uncertainty in that estimate, well, we add the variances. So the variance of the indirect estimate is the sum of the variance of the direct estimates that we've used to make that indirect comparison. So we're getting, it's going to be less certain than any of the individual direct estimates itself. And I think that's reasonable because we're making an indirect comparison. We're not getting a direct comparison. But on the basis of that, the committee looked at that evidence and they decided that actually it did seem that they were similarly effective and that it represented good value for money, and so they recommended it on the basis of the indirect comparison. So that's a simple example of network meta-analysis, but they can be much more complicated. So we could have both indirect and direct evidence, like the triangle on the left. So there we can, or we could just, if we only focus on treatments A and B, we could just use the direct trial that compares B and A. But if C was also an option in our decision problem, we want to make decisions between A, B, and C, then we need to combine all that evidence. There's no reason, why would you pick the AC evidence to look at, or BC, or the AB? So we could pull all of the evidence together to be able to get a pooled set of estimates which compares these three treatments. And what we want to estimate there are just two relative effects, because with three treatments, there are two relative effects that we need to estimate. So we've got three bits of information and two parameters. This is the graph in the middle is showing some studies which compared thrombolytic treatments for patients who've had a heart attack. And there you can see we've got some sort of spurs of evidence which will just be informed by the direct estimates. We've also got some loops of evidence where we'll get an average of the direct and the indirect evidence. And effectively, the evidence sort of flows around these networks to be able to give us a pooled set of estimates. There's a nice analogy that Gertrude put forward, that you can think of these as like electrical circuits. And so you've got evidence flowing around this electrical circuit. And if you've got more evidence, then the flows better, and if you've got less evidence, it flows less well. And I think the analogy works quite well. One thing you do need, however, is a connected network. So if you have studies, let's say you had an ED study on the left-hand side there, because there's no links to the triangle, we can't then compare the treatments in the triangle, the ABC, with the DV. We can only make comparisons within a connected network. And on the right there, there's just to show you that these can be very complex. This is an example of treatments or interventions for social anxiety. And if you define the interventions very precisely, then you get this very big, complex network. Although you can simplify that by considering those interventions belong in intervention classes. Okay, you can see there that telling if it's connected or not isn't necessarily straightforward. Okay, so how do we do this? Well, it uses that same idea, that simple maths that I talked about for the indirect comparison. So we've got how many treatments that we want to get estimates for. So we need, in terms of relative effects, we've got as many relative effects as there are treatments minus one, because everything is relative to treatment A. So we estimate the treatment effect of B against A, C against A, and so on. And then for any studies that tell us about any other contrasts, say a study that told us about B versus D, well, we use that consistency relationship that I described, that the effect of D against B will be the effect of D against A, take away the effect of B against A, and so on. So the studies which tell us directly about one of our relative effects that we want to estimate, sort of the basic parameters, they're studies against treatment A that got an A arm. But any studies that don't have an A arm tell us about these functions of parameters. I guess that's why these methods were developed using Bayesian methods, because there weren't any off-the-shelf frequentist approaches to be able to estimate such methods, models. Whereas in a Bayesian approach, you can just write out your likelihood, you write out your functional relationships and your links, and then files, and then you can just get it. So I suppose I've come into being a Bayesian through the simplicity or the ease by which I can make up or think about models that I want to fit, and then I can just write them down and fit them. You don't need to find off-the-shelf software that will do that. So just to note that the model will then estimate estimates which are consistent. So it means that whichever way you look at it, the conclusions you draw will be the same. But the data may not be consistent. So it could be that your A-B evidence won't be telling you exactly the same thing as the indirect estimates. If in our triangle, if our A, C and B, C data gave us one estimate of the B against A effect, then our direct data gave us a different estimate, then we've got some inconsistency there between what the study of B against A is telling us and what the indirect evidence is telling us. And if that happens, then well, that's a problem for decision making, because what do you believe? But it really means that you need to go back and look at your data carefully. It could be that, say, as a good example is if, which way round? If C was surgery, then A against C would have to be on a population of patients who were fit enough to have surgery. And so would the B against C studies, because they include C, they include the surgery option. But the B against A studies don't include the surgery option, so they could be on a different population potentially of people that perhaps weren't so fit, and so may have less good outcomes, and they may do less well on these treatments. So we need to think very carefully about what's going into our evidence synthesis and only put together things which are on effectively the same population and making the same comparisons. Okay. I was told that it's okay to put equations in, so I put some equations in. So if we had binary outcome data, oh, I've got this again.
Here it is again.
It must be in my presentation. Okay. So yeah, if we have binary outcome data, then we would naturally put a binomial likelihood on our arm level data from our studies for study I, arm K. So we can have studies that have got up to as many arms as you like, actually. It could be 3, 4, 5 arms. And they estimate some probability of effect P, the probability of, I'm so sorry because it's pointing at the wrong thing. Ignore the little dot. So we're estimating a probability of having the outcome P on study I, arm K. And a natural thing to do with binary outcomes when you've got probabilities is to model that on the logit scale. So if we use a log odds link, then on arm 1, we're going to estimate the log odds of having the event mu. We don't really care about that, it's just a nuisance parameter. We give it a flat file, we've got to estimate it, but it's not something of relevance to our problem. What we're interested in are the relative effects. So that's the deltas, which is the treatment effect of the treatment that's on arm K in study I compared with the treatment on arm 1 of study I. So to be a bit careful here, because arm 1 might be different in the different studies. It's a mu represents something different in each study. So how do we make sure we make the right comparison from that study? So those deltas, that's the treatment effect of study I, arm K. Well, that's equal to the treatment effect for TIK. So that's the treatment that was on arm K of study I. So if it was a D versus A study, then that would be the treatment D, it'd be the treatment effect for treatment D. And that's compared with the treatment effect on the first arm of that particular study. So basically pulling out the different treatments, it's like a lookup to tell us what treatment comparison that particular study is making. And then we make sure that we will use the right consistency equation there for that particular study. I tend to give flat priors for things. I know we can discuss this and you'd be very welcome to do something different. But especially the news, they're nuisance parameters. I don't want to bias my results at all by letting them be anything different than they need to be. So I would just give a flat prior for the news, and I generally give a flat prior for the treatment effects because we can usually estimate those quite well from the data. Although exceptions might be if you've got sparse outcome data like adverse events and so on. Now we can either fit sort of a common effect model that's often called a fixed effect model, or a random effect model. So that's where, for a common effect model, then we assume that the treatment effects are the same in all studies that, sorry, I'll say that again. So in a common effect model, we assume that all studies making the same comparison are estimating the same true effect. The fact that we get different results is just due to sampling error. Whereas in the random effect model, we assume that all studies making the same comparison would get different effects because there are some differences between those studies on the populations, on the interventions, on the way that the study was conducted. And then we estimated between study variance parameter, that's this tau squared. You need to have quite a lot of studies to be able to estimate this, but if you do, you can estimate this random effects model, and then you get an estimate of heterogeneity between studies. I put a uniform multiplier here. I was illustrating the multi-NMA package to estimate this, which doesn't like bounded priors, and in that case, you might use something like a half normal to make sure that that's positive. Okay, so treatments for, so this is an example of those thrombolytic treatments that I mentioned. So these are various different thrombolytic treatments for patients who've had a myocardial infarction, and this is the network plot of data that we have available. If we fit that model, then we can then get estimates of the log-odds ratio for each of those treatments versus streptokinase. And these will have the property that they adhere to those consistency equations. So if we took any two of these, we could take the difference and then we'd find the other. And we can see from this plot here, so 0 would be no effect, that there's reasonably clear evidence that PCTA, which is actually not a drug, it's a angioplasty, it's like a little balloon of blood in your veins to try and increase, to get the blood flow, remove blockages. You can see that seems to be more effective than these thrombolytic treatments. I mentioned that we've got to check that our evidence is consistent. So we can do that. We can have a look to see whether or not the estimates for each treatment from the direct evidence on that particular treatment agrees with the estimates based on the rest of the network. So we do these one at a time. We take each contrast one at a time. And we can then fit a model which sort of, it's called a node splitting approach, where we take out one treatment comparison, estimate it using the direct data, and then use the rest of the network to find an indirect estimate. And then we can combine that, we can compare that together, and we can also compare it with the overall network meta-analysis estimate. And that's what's being shown here for each of these comparisons. You can see that well, for a start, the network meta-analysis of the blue bit is more certain because we're pooling both direct and indirect evidence together. And on the whole here, we've got overlap between our direct and indirect estimates. And so there isn't really any cause here to worry that we might have any inconsistency in these estimates. I showed an example yesterday for the same set of data, but when some older treatments were included in the network as well. And there we did see some inconsistency because those older studies were somewhat different and they've been done a long time ago. So you need to be very careful about what you put together. Okay. So that's my quick sort of whistle-top introduction to network meta-analysis. But this is a Bayesian conference. So as I was putting this together, I started to think to myself, well, why take a Bayesian approach? Historically, it's been done certainly in decision-making context because there was a need for a method to be able to do this, and there wasn't anything in the frequentist world to do it, and we could easily write this to do it. But these days, actually, there are a whole range of frequentist packages which will also do network meta-analysis, and to be honest, you'll generally get similar results. Differences are that they don't treat uncertainty in the between study variance properly, so they assume that's known and fixed, and so you'll get more precise estimates, but on the other hand, it's not properly reflecting the full uncertainty of the evidence. So there are some differences. But really, I think that if you were just doing a very simple network meta-analysis, there's probably very little difference in the two approaches. But it still is the case that if you want to do anything slightly different or slightly more exciting or more complex, then there isn't, you can only go so far in the frequentist world with the, what's in those packages. Whereas if you're working in, if you want to do something more exciting, then really you do have to then, well, for me anyway, I can just, I want my likelihood to look like this, I want to be able to model this, I want to add in these parameters, and I can just write them out in my code, and then off it goes, and I can fit it. So I suppose that's what I think, that's where I come from in terms of being a Bayesian. It allows me the flexibility to do the exciting modelling I want to do. And so I'm going to now take you through a few more exciting bits of modelling for network meta-analysis. Okay, well, I suppose firstly there's this focus on decision modelling. I'm not just looking at getting treatment effect estimates. I want those estimates to be fed right through an economic model, which might be quite complex. It could be a Markov model, it could be a discrete event simulation model, and it might have loads of inputs going into it, and all those inputs are uncertain. And I want these estimates from my network meta-analysis to be able to feed into that. And it's really easy to do that if you've done a Monte Carlo Markov chain simulation, because your simulated values are what you've already got, the simulated values that you need to feed into that economic model. So what they tend to produce in decision modelling are these cost effectiveness planes. So on the x-axis here you've got the difference in effects, so your benefits of the treatment, and on the y-axis you have the difference in costs. And you want your scatter of points to be sort of in the bottom right scape, so you're getting more benefit for cost. So below that threshold line, so anything to the left of that threshold line is considered not cost effective, and anything to the right of the line is considered cost effective. So network meta-analysis allows you to, so when you fit a network meta-analysis, your estimates will be correlated with each other because they've been fitted in a joint model. And we need that correlation to be propagated right through into the economic model. The economic model also will be based on some quite complex functions of your parameters. In the simplest case, you just want to get back to a probability scale, but you need to apply your relative effects, your log odds ratios, to some baseline log odds for the model to be able to get the probabilities of outcome in the model. So again, MCMC simulation is really easy because you just have to have one line of code which basically puts that equation there, and you've got the probabilities you need, and they can be propagated directly through the model. Okay, another example is when we're estimating the between study variance. Very often we know there's heterogeneity between studies, and we would expect it, but we don't have enough studies with which to estimate it. So Gelman in 2006 said that you need at least 4 or 5 trials in a comparison to be able to adequately estimate that. That's probably an underestimate. You probably need more like 10 or 20, because I mentioned this yesterday, that really the number of studies you've got is your data set for estimating between study variance. So some work by Becky Turner and Kirsty Rhodes went through a whole big database of past Cochrane reviews to be able to estimate distributions of between study variance parameters from previous meta-analyses. And they based that on the outcome type, what sort of intervention comparison it was, what type of clinical area it was. And they used the sort of meta-analysis to be able to make predictive distributions for what the between study variance might be in a new meta-analysis. They can then be used as an informative prior or an evidence-based prior for the between study SC. So just this is an example, we use this in the NICE clinical guideline for post-traumatic stress disorder in children and young people. So this was a review which was looking at non-pharmacological interventions with a continuous outcome. And it was in mental health. So we picked out the relevant informative prior, which is the one on the right there, which was based on this big analysis of previous meta-analyses to give us a prior for between study SD. I realise I haven't plotted these on the same scale, so it's not so easy to compare, but the one on the left is if you just use an informative prior for SD. We really didn't have enough studies to estimate it. But on the right, we use our informative prior, and then it brings in the estimate much more tightly, so we get much more estimating power if we use this informative prior. So another thing which we do quite a lot actually, because we support the NICE guidelines work in Bristol, and very often they have data that's been reported in a whole range of different formats. They still want to be able to combine all that data together. I think meta-analysis is very often have lots and lots of different analyses for different outcome reports, formats. But actually, really, these are all trying to estimate the same underlying parameters. It's just they report them in different formats. So analysing the separate things and splitting the studies up is kind of losing something. What you're not doing is bringing it all together to be able to get an overall estimate for the underlying parameters of interest. And if there's functional relationships between what's been reported, then we can actually write that down to a formula, and then we can estimate it very easily using a Bayesian approach. So, for example, some studies might report event rate data as a binomial likelihood, so it could be the number of patients who have an event after a follow-up time t. Others may report it as the number of events for a certain person years at risk, and then you'd have a plus on likelihood. But they're actually estimating the same underlying parameters. So other examples are where you have sort of these collapsed category data. So some studies might report results for stage 3 and stage 4 patients, whereas others might actually have a mix of those in their results, and they might tell you the proportion of stage 3 patients, which is effectively a weighted average of the other types of data. And so you can write down that relationship and you can estimate a joint underlying model. I'm a bit worried about time. I've got loads of examples, I can keep going through them, but tell me when to stop.
You have 5 minutes.
Okay, all right. So this is an example of that sort of shared parameter model, where we had studies here on hip replacements. And in our model, we had a piecewise continuous model. So there were 2 periods of time. And, there's a sort of a different revision rate early on. So when you just had the operation, then there may be quite a high revision rate if it hasn't been successful. But if it's been successful, then it's actually usually quite a long time before revisions start to happen again, because you need to wait for the joint to wear out. So we had 2 periods, a red period and a blue period, where we were estimating the relative effects on the hazard ratios. But different studies reported at different time points. Some of them reported at multiple time points. We had a study here that reported firstly in the first period, and then again in the second period. Well, there's 3 sort of pieces of data that they're reporting there in terms of revisions, and they've had different exposure periods within the different intervals. So in the first period, that's all the first parameter, the revision rate in the first period. But in the second period, you've got a mixture. You've got a sort of a weighted average of time of exposure in the first period and time of exposure in the second period. So we can write that out. We've got binomial data. We can link that to a hazard ratio through this relationship between the probabilities and the rates. And then we put our model on the rates. The rate is a weighted average of the time spent, the exposure time in each of those 2 periods. And then we can have our network meta-analysis on the log of the rates in those 2 periods. So different studies would tell us about different times and piece it together. But we can estimate these models if it's straightforward. Yeah. If you can write it down, you can estimate it. That's what it's like. This is just showing the number of studies that were reported in that first period and those in the second period. And then we can get estimates out for each of the interventions at the 2 different time periods. Okay. So I'm running out of time. So I won't say very much about this, but this is you can also use these models to fit bias adjustment, and you can adjust for biases and potential biases in studies, either again using evidence-based pliers or maybe using elicitation from experts and work by Becky Turner on that. Or you can try and estimate it internally within your NMA using studies that are deemed to be at high or low risk of bias. And with small study effects, which are sort of often explaining sort of publication biases, then you could potentially fit regression models. I'm not going to show you the maths, but we did this in a review of different forms of fluoride into for children to prevent fillings in your teeth. And what we found was the blue lines here are the effects if you don't adjust for bias, whereas the blue and the green lines are different ways of adjusting for bias due to small study effects. And the first thing you notice is that it moves the estimates closer to the null if you adjust for bias, and that is particularly the case in the studies which looked at giving fluoride within the varnish, which then brought the fluoride effects much more in line with each other. The main conclusion for this is it's a good idea to brush your teeth. But it was interesting because the placebo is better than no treatment, and that's because placebo was brushing your teeth with paste that didn't have fluoride in it. So it's actually quite an active intervention. You are actually brushing, you're doing quite a lot of good, it's just you don't have the fluoride. Okay. I've run out of time to talk about this. I'm just, there were some questions yesterday about what to do if there's differences in populations and adjusting for that, and if you have single arm studies or and you want to try and bring those in and try and adjust for potential biases. And this network meta regression model is an approach to do that, and it can be fitted in the multi-NMA software that I showed you yesterday. It requires some quite complex functions, but again, they're just functions you can write down in an estimate. But I don't have time to talk about it, and it requires quite a bit of time to talk about. So I'm just going to leave that there as, yeah, there's more further exciting things that you can do. And I should probably skip to my last slide at that point. So yeah, I think network meta-analysis is great for being able to make the decisions that we want to do. It doesn't really need Bayesian methods for the simpler approaches, but as soon as you want to do anything more exciting, then Bayesian approach, I think, is essential. And there are lots of challenges ahead, and I think in particular dealing with disconnected evidence networks and single arm studies, and I think that's a really active area for future work, and I think Bayesian methods are really important role.